Hey guys, welcome to Medifaction. So today let's learn about some physiology of glucagon. Glucagon is a hormone that works with other hormones and bodily functions to control glucose levels in the blood. So, to learn more about glucagon, let's begin. In this video, I'm going to enumerate an introduction, source of secretion of glucagon, chemistry and half-life, synthesis, metabolism and actions of glucagon. We are also going to discuss about the mode of action and finally we will learn about the regulation of glucagon. Introduction. Glucagon is a peptide hormone produced by alpha cells of the pancreas. It works to raise the concentration of glucose and fatty acids in the bloodstream and is considered to be the main catabolic hormone of the body. It is also used as a medication to treat a number of health conditions. Source of secretion. Glucagon is secreted from A cells or alpha cells in the islets of Langerhans of pancreas. It is also secreted from A cells of stomach and L cells of intestine. Chemistry and half-life. Glucagon is a polypeptide with a molecular weight of 3485. It contains 29 amino acids. Half-life of glucagon is 3 to 6 minutes. Synthesis. Glucagon is synthesized from the pre-prohormone precursor called pre-proglucagon in the A cells of islets. Pre-proglucagon is converted into proglucagon, which gives rise to glucagon. Metabolism. About 30% of glucagon is degraded in liver and 20% in kidney. The cleaved glucagon fragments are excreted through urine. 50% of the circulating glucagon is degraded in blood itself by enzymes such as serine and cysteine proteases. Actions of glucagon. Actions of glucagon are antagonistic to those of insulin. It increases the blood glucose level, peripheral utilization of lipids, and the conversion of proteins into glucose. Now let's go on each. Number 1. On carbohydrate metabolism. Glucagon increases the blood glucose level by number 1. Increasing glycogenolysis in liver and releasing glucose from the liver cells into the blood. Glucagon does not induce glycogenolysis in muscle. Number 2. Increasing gluconeogenesis in liver. But how? A. Activating the enzymes which convert pyruvate into phosphoenol pyruvate. B. Increasing the transport of amino acids into the liver cells. The amino acids are utilized for glucose formation. Number 2. On protein metabolism. Glucagon increases the transport of amino acids into liver cells. The amino acids are utilized for gluconeogenesis. 3. On fat metabolism. Glucagon shows lipolytic and ketogenic actions. It increases lipolysis by increasing the release of free fatty acids from adipose tissue and making them available for peripheral utilization. The lipolytic activity of glucagon in turn promotes ketogenesis, that is, formation of ketone bodies in liver. Number 4. Other actions. Glucagon. It inhibits the secretion of gastric juice. It also increases the secretion of bile from liver. Now let's see the mode of action of glucagon. On the target cells, mostly liver cells, glucagon combines with receptors and activates adenyl cyclase via G protein. Adenyl cyclase causes the formation of cyclic adenosine monophosphate, which brings out the action of glucagon. Glucagon receptor is a peptide with a molecular weight of 62,000. So this is the diagrammatic representation of physiological action of glucagon. Glucagon, on glucose, it increases the blood glucose level. On fats, it decreases lipogenesis. On insulin, stimulates insulin secretion. Now let's see the regulation of glucagon secretion. Secretion of glucagon is controlled mainly by glucose and amino acid level in the blood. Number 1. Role of blood glucose level. Important factor that regulates the secretion of glucagon is the decrease in blood glucose level. When blood glucose level decreases below 80 mg per deciliter of blood, alpha cells of islets of Langerhans are stimulated and more glucagon is released. Glucagon in turn increases the blood glucose level. On the other hand, when blood glucose level increases, alpha cells are inhibited and the secretion of glucagon decreases. Role of amino acid level in blood. Increase in amino acid level in blood stimulates the secretion of glucagon. Glucagon in turn converts the amino acids into glucose. Role of other factors. Factors which increase glucagon secretion. Number 1, exercise. Number 2, stress. Number 3, gastrin. Number 4, cholecystokinin. Number 5, cortisol. The factors which inhibit the glucagon secretion are number 1, somatostatin, number 2, insulin, number 3, free fatty acids, number 4, ketone. So this is an easy diagrammatic flowchart for identifying the glucagon functions. On glucose metabolism, it increases glycolysis and it also increases gluconeogenesis. On lipid metabolism, it increases lipolysis and as well as it increases ketogenesis. On protein metabolism, it increases ureagenesis and it also increases hepatic amino acid uptake. In case of energy metabolism, it decreases the food intake but it increases energy expenditure. Hope you have understood the video. Like, subscribe and press the bell button for more videos. Thank you. Thanks for watching.